Here we are in the fourth topic of discussion, ladies and gentlemen. As I promised to you that today will be very fruitful and will be very inspiring for all ladies and gentlemen out there, not only in this region but around the world. This fourth topic of discussion is very interesting because it is revolving around PharmVIP, a one-stop platform for pharmacogenomic variant analysis and interpretation that will be delivered by the representative from National Center for Genetic Engineering and Biotechnology. It is our greatest honor and pleasure, ladies and gentlemen, to have Dr. Chittima Piriyapongsa with us here.
Good afternoon, everyone. My name is Chittima Piriyapongsa. I'm the team leader of Data Bank Research Team at National Biobank of Thailand. Today, I want to introduce a new pharmacogenomic software platform called PharmVIP, which I want to develop it to be as a one-stop platform for analysis and interpretation of genetic variants in all pharmacogenes. The name PharmVIP is from the function of the platform I mentioned earlier, Pharmacogenomics Variant Analysis and Interpretation Platform. So the motivation for developing this platform is due to some pain points that pharmacogenomics researchers currently face in variance data analysis. So these are main features of PharmVIP which can solve these limitations. First, in PharmVIP, which have 3 analysis modules, researchers can comprehensively analyze all pharmacogenes in various aspects within one tool, so no need of using multiple tools for variance analysis. Secondly, this platform is easy to use even by users unfamiliar with data analysis or command line tools, because the analysis can be done through a web interface by only selection and clicking buttons, so no command line, no programming. Also, the output report will be presented in easy to understand format, both in summary report and full report, which I will show later. And lastly, this platform is designed to be able to accept large input files, which are usually limited for a web tool. PharmVIP basically consists of 3 analysis modules, as I mentioned before. First, the guideline modules is provided for allele prediction in genes which have information of CPIC drug dosing guidelines. So these include the gene-drug pairs with strong association between genotypes and drug response phenotypes, such as the gene CYP26, CYP2C19. Currently, these modules predict alleles in 17 genes associated with 47 drugs. So this module accepts a WZIP file as input. And if user want to analyze CYP26 as well, the BAM file is also required. So the platform will analyze the variants and haplotype information and reports the possible dipotypes of each gene and the associated phenotypes, metabolizer status, and the corresponding CPIC recommendation. The second module is for HLA allele prediction. So this module predicts alleles for all HLA genes from an input BAM file using different methods, and then reports the possible HLA alleles in all genes with the list of the drugs and adverse drug reactions associated with these alleles, which were reported in the published studies. The report also give the details of the published studies and the corresponding CPIC guideline if available. The third module is pharmacogenes modules, which focus on the identification of variants such as the SNP, insertion, deletions in all pharmacogenes from an input VSEP file, and then predict the effect of these variants to gene function, such as the variant class, types, consequence, and impact level. This slide displays the rough workflow underlying these analysis modules. So in brief, for guideline module, we use our own scripts to pre-process the input data, perform allele matching, and assign CPIC guideline to the matched alleles based on the information of alleles and guidelines from the PharmGKB database. For HLA, we use several methods to predict HLA alleles from input BAM file and report the summarized consensus results, including the matching alleles to the associated ADRs that have been reported. For pharmacogenes module, we use variant effect predictor software to assign the consequence or effects of identified variants to the gene function. And after analysis is finished and users see the results, users have an option to filter the results again to get only the results fit with their interest in both summary and full report format. So you can see the advantage of PharmVIP here, that users don't need to understand or learn how to implement all these workflows by themselves, but can easily use them through a simple web interface. Now let's see the PharmVIP website, and I will show you an example of how to use it. When user go to the web, we'll see the page home like this. And first of all, user has to create an account by click sign up here. Having an account is useful because user can use the account to manage their own projects and can always log in to check the analysis status and analysis report of the project. And after the registration is confirmed by an email, user can click to sign in here. Sign in. And to start a new project, click on Dashboard. And then click on the New Project button. And put on the name of the project. I'm going to put test 2 here. And user also can put some additional details of the sample here, which is optional, and then click the Create Project button. So after you have created a new project, you can see that the project show up in the dashboard at the status saying waiting for an input. To start analysis, user can click on the Start button of the project that you want to run an analysis here. And the platform will take you to follow 4 analysis steps. For step 1, users choose an analysis option out of 3 modules that I mentioned before: the guideline module, HLA, and pharmacogenes. So now I will select all modules here. The 3 analyses will run in parallel. So it will go to step 2 for uploading the input files for each selected module in BAM or VSEP file format. I will upload the VSEP file for the guideline and pharmacogenes modules. And I'm gonna put BAM file for the CYP2D6 and HLA modules. For step 3, select the analysis parameters for each module. User can simply use the default which analysis or the genes here, or can select specific list to be analyzed. For the guideline assignment module, user can choose to analyze from the gene list or the dark list. So I'm going to show select by gene list. There are 17 genes now, or user can select from the drug that group by therapeutic category here. You can see for each group, you can select the list of the drug or select for the whole group here. And now just say GI, neuro. So you can select specific drug here that you are interested to analyze. I'm going to select for all the genes. And you can also set the platform to report only the best candidate allele in case that more than one allele is predicted from the platform. And for the HLA allele prediction module, it will analyze all the HLA genes. And in case user focus on the associated adverse drug reactions that found in specific population, user can choose the specific ethnicity of the cohort which reported ADR cases associated with the predicted HLA allele. And for variant identification in pharmacogene module, user can choose the list of pharmacogenes to be analyzed here. So the list of pharmacogenes here are from different sources, such as some publication or the database, such as the drug bank, PharmGKB, CP guideline, or some specific genotyping platform. You can select all here, or just some lists. In addition to within gene analysis, user can specify the boundary of up and downstream regions that user want to identify the variants as well. So after finish parameter selection, you can click Save here. Then it comes to the last step. User can check the correctness of parameters and go back to change it if you want. To begin analysis, click Start Analysis here. User can check the status of the project on the dashboard as is shown here. Queuing, and then running, and the percent progress of each module will display here. You can see it's continue to increase. So if you fast forward the waiting time here, so I can finish it in time. The running time is about 24 minutes. So when the analysis is done, user can click on the result button here to check all the results and the report. Let's see an example of the summary report. So you can look at the summary presented in interactive web version or can export it in the PDF file format. So let's take a look at the web version. So the report will start by the project details here and the option and parameters of analysis that user has selected. Then the summary of each module, starting by the first module, guideline. So the first table will show the type assignment of each gene. So for each line, it will tell you the name of the genes, the phase status of the genotype data, and the number of missing variants in the haplotype, the number of the diplotype that can be predicted from the genotype, and then the list of the matched diplotype. You can click on the link to see the details of the diplotype. For each line, we'll tell you the detail of each variant, the RSID, the related alleles with this variant, and then the sample genotype from the input file, and the haplotype number 1 and haplotype number 2 combined to be the diplotype *1*1 here, and also the read depth information. So this tells you how many reads support the assigned genotype here, and can help the user to determine the confidence of genotype of each allele as well. So let's go back. And the second table will show the related CP guideline of the predicted diplotype. For example, the CYP2C19 here related to the drug citalopram, and the diplotype *1, *2 was predicted for these genes, which related to the intermediate metabolizer, and recommend to initiate therapy with recommended starting dose, and this guideline is supported by strong evidence. So you can click here to see the detail of the genes from the PharmGKB website. Also can click on the link to see the drug information and the related pathway. And there's a link to the full report as well. The second module, the HLA. The first table will tell you the detail of the predicted allele of all the HLA genes, both the class 1 and the class 2. And for each line, will tell you the predicted allele and the number of methods out of the total 3 methods that we use that can detect this predicted allele. Also, the ADR link to the table below, which have the detail of the associated ADR. So I come back here. For the second table of the HLA, will show the related CP guideline, the same as in the guideline module. And the third table will show the list of the HLA-ADR association study that related to the predicted allele. For example, this one, each line will tell you the drug name that related to the ADR and the disease of the patient of a reported case of ADR, the population, and the number of publications related to this event. And it's also linked to the full report, which will give the full details of the study. And the last module, pharmacogenes, the summary will show in the form of summary statistic of all the variants identified in the sample. So this graph shows the summary of the variant classes. Most are SNP, and there's some deletion and insertion as well. Also, the distribution of the type, novel and known variant, and the consequence, basically the effect of the variants to the gene function. This one most are intron variants in blue color, and the impact level. The blue one here is a modifier, and there are some part of it have high impact, 0.08% here in red color. And the effect to the coding sequence, most are missense variant here. And also the result from the shift and prevent software. So this is the summary in the web version, or it can be exported into PDF file here. So the report will be in a nice and easy to understand format. So it's useful actually for the user to interpret the analysis result. So if user want to see more details, can check on the full report of each module. So let's see the guideline module. So the full report will show the details of each CP guideline here. For HLA, it will show the similar result to the summary report plus the details of the association study related to each predicted allele. For example, here you can click on the publication that reported ADR cases. And for gene module report, there's two wheel option, the variance wheel and the gene wheel. So for the variant tab, we'll show the list of all identified variants with the details, which can be exported in an Excel file here. And for the gene tab, we'll group the variant by the genes and let the user see the summary of variance location along the genes in the lollipop. I will display here. So each color here represents the different variant effect to the gene function. Like in the red color here is the stop codon, and this one is frame shift. The yellow is missense, and the blue one is the synonymous. So let's go back. So after checking the report, user can filter the result in each module here separately to get a new report with only result of interest. For example, I want to filter the result in the guideline module. So I try select only the CYP2C19 gene with the phenotype intermediate metabolizer, and select only the guideline with strong support evidence. And then I click filter here. So let's check the new summary. The result will be updated accordingly to the filtering criteria that we use. It shows only the CYP2C19 here with the intermediate metabolizer and only the guideline with strong supporting evidence. You can see that this filtering option is unique and quite useful actually. Users can generate reports again and again to fit their requirement without the need to rerun the analysis. So these are examples of PharmVIP function and how to use it in brief. So in summary, this integrated PharmVIP platform is useful in identification of variants and alleles for all pharmacogenes and interpretation of their consequence in terms of gene function, the associated adverse drug reactions, drug response, and also the dosing guideline. So for the current status of the platform, now PharmVIP is unpublished work, so the web URL is still not public, but we plan to publish the paper soon. Now the platform is in beta version, so there's still some room for development further which will require feedback from all the users as well. And we expect to add more features to the next version of PharmVIP. For now, the platform will be provided for research and informational purpose only, and it's not meant to be a substitute for medical advice by a doctor. Finally, here are the PharmVIP development team from National Biobank of Thailand. So from the presentation today, I hope PharmVIP will be a new platform for variant analysis and interpretation that can be useful to the pharmacogenomic community. If you have questions, suggestions, or specific requests, such as to use the PharmVIP platform, you can send an email to me at this address. And thank you for your attention. And if you have questions now, I'd be happy to answer them.
Thank you very much, Dr. Chitima, as well as your distinguished team members from National Center for Genetic Engineering and Biotechnology as well. Thank you very much. I believe that they have learned a lot from your sharing, and our participants would like also to take part in the discussion further on the topic as well. So there are some questions from the floor, and I would like to ask for your kind participation in taking some of your time in answering all of them, please.
Can you hear me? Anyone hear me? Okay. Thank you. Thank you for listening. And now is the Q&A sessions. So I got two questions here. So what are the difference between PharmVIP and PharmCat? So for PharmVIP, we have 3 analysis modules as I show in the presentation. So I would like it to be like a one-stop platform that can do several aspects of the analysis, not just providing the CP guideline, but PharmCat focus on the giving the CP guideline related to the some genes that have CP dosing guideline only, but we plan for this to be able to analyze several aspects like the guideline, HLA, and also the variance identification and prediction of the effect of the variance as well. And for the guideline part, compared to the PharmCat, I think the main step should be similar for both PharmVIP and PharmCat. There is a type matching, the guideline assignment, but there's some difference in the details. For example, we add some lead depth information so the user can determine the confidence of the genotype calling of each position. Also, we got consistent results to the PharmCat for majority of the cases, but still there are some cases that we miss, and while some cases PharmCat miss. So we will determine the reason and handle all those cases. So we will need a lot of test datasets in order to identify those cases, especially the dataset from other experiment tests that know the real answer, since there's not many gold standard dataset available out there. So if anyone can contribute this kind of data, I would highly appreciate if you could. I also would like to thank you, Professor Watson, for giving me the opportunity to present the tool and also offer to provide the dataset to test the software in the future as well. And for the second question, I just got, is there any plans on making PharmVIP downloadable for offline use? Yes, we plan to have a different version. In addition to the online version, we would like to make the local version that you can run on your computer as well. But for the main objective, for the initial objective for developing this tool is for the user that unfamiliar with the data analysis, which I think is the pain point of several users for the pharmacogenomics community now. Third question, what is the future plan for PharmVIP? Will it be commercialized, or I have not thought about that, but for now, since currently we intend for the research and informational purpose, so it's free for now, and it's certainly free for the academic use. Is there any more questions? Okay, no questions. So if you have any more comment or suggestion or anything, you can email me at the email address I showed in the presentation. So I would like to get all the feedback as much as possible that I could, so I can improve the tools to fit with the user needs. Thank you.